Hello, everyone. So, today I'm going to discuss the experiment in the pharmacognosy course, and the title of this experiment is Morphology, Histology, and Powder Study of Ginger. So, the outcome of this experiment is at the end of the experiment, you will be able to identify the various morphological and microscopical diagnostic features of the ginger. Now, coming to the requirement for this experiment, since these experiments involve the morphological, microscopical, and powder microscopy, so we need, first of all, the fresh ginger rhizomes. Then we need powder of it, and we need magnifying glasses for the morphological, especially. Then we need the microscope, especially the digital microscope, and we need watch glass and slides with cover slip, dissecting needles, and we need brush to transfer the section, and then after cutting it, and then blades for section cutting. And we are using blades for section cutting, but there are instruments which are used for the fine section cutting in very sophisticated manner, like microtome section cuttings. uh, then proper forceps, and in chemicals we need fluoroguanol, concentrated HCl, dilute iodine solution, and sudanrate. So these reagents we need to use for identifying the various microchemical structures in the TS. Now, coming to the background of these experiments, so first of all, we should know why we are doing it. What's the purpose of doing this experiment? I think you already studied this in the theoretical part where we learn about the adulteration, and we discuss about how this adulteration happen in the commercial market for the drugs which are having the therapeutic value or commercial value. And this adulteration is solved by various evaluation methods, and in Haba industry they got the various evaluation methods to check for this adulteration. So this adulteration can happen because of many reasons, and we already learned in theoretical part of this topic, wherein we found that some of the adulterations are done unknowingly, some are done knowingly to earn profits, some may happen accidentally because of the uh, unskilled workers who collect the plant from the fields during the harvesting, or adulteration chances are there during the processing and storage. So this adulteration can be checked by various methods, and this is why this is where our practical comes in picture. So there are various evaluation methods to check for the adulteration, and today we are going to use a very basic method for checking the adulteration, that is morphology and microscopy. Of course, there are other evaluation methods, as we learn in the evaluation topic. Like, we can even use the physicochemical methods for checking the adulteration, and we also need to use the chemical methods, and we have various chromatographic techniques. We have got various spectroscopic methods to check for the evaluation, or to do the evaluation of these adulterations, to check the adulterations. So now this morphology and microscopy is a very basic, you can say, method which was used in the very beginning, when there was no much scientific instruments or very sophisticated methods available. So the herbalist, or the people who used to deal with the herbal medicine, they used to identify the authentic drug by using the sense organs, by smell, taste, touch. And of course, after the advent of this microscopic techniques, there was some of the microscopic methods which was brought, like simple microscope and compound microscope. They were used to identify the microscopic structures, to detect or to identify the very close species of these adulterated plants, or drugs in the market. So we have a sample of ginger, and if you look at the background of the ginger, it has, the botanical source, or we call the scientific name for the ginger is Gingiber officinale. And if you look at the source, it is obtained from the dried rhizomes of the Gingiber officinale, name of the taxonomist here. And then family is Gingiberaceae. So there is a difference between the rhizomes and roots. Okay, so rhizomes are modified stems. Okay, and if you look at the chemical constituents, important chemicals, ginger contains the oleoresins. That means with resins with the volatile oil. So we got volatile oil. We got volatile oil together with the resin. So that's why it is called as oleoresin. And it has the principle like gingerol, gingiberine, gingerone. So gingerol is actually the pungent principle in the ginger. So we know the ginger got a lot of medicinal properties. So these medicinal properties are because of the presence of these uh, chemical constituents. So this chemical constituents, these three are not limited. The list here is not limited to these three. There are many other chemical which are present in ginger, including the starch and many other types of the chemical constituents are present in ginger. But we have captured here the main one. And then coming to the uses, ginger, as we know, it is used as carminative flavoring agent. So there are so many products where you will find the ginger flavor, maybe candies. You will find a lot of food products, a lot of the edible products which has the flavor of ginger. Then it has also been used in nutraceuticals. You will find so many ginger based nutraceuticals, and also you will find the ginger as one of the very important herb used in food, especially in cooking. So if you look at the ginger genus, as you can see here in this botanical name, gingiber is a genus, officinale is a species, and rossuk is actually the name of the taxonomist. So if you look at the ginger genus, it may have so many other ginger varieties. As we know, there is a Japanese ginger, there is a Jamaican ginger, African ginger, we know the ginger from Indonesia, ginger from India. So we have got so many varieties of the ginger, and in commercial market you will find quite many number of the varieties of the ginger. And the genus of all these gingers remain the same, that is gingiber. An official variety which has the medicinal property is actually officinale, but there are possibilities where in the very close varieties or species which doesn't have those chemicals, these oleoresin chemicals which are important for the therapeutic purpose, may not be present, or you can say may be present in a very less quantity. So they are sometimes used for adulteration as well. So ginger genus may have a similar varieties with a similar morphology, but they have a different pharmacological and therapeutic properties. So there is a need to check for those adulterations. And as I mentioned just now, that it is used for cooking and also having medicinal properties. So the authenticity of the ginger in the market, it's very, very important for the food industry and also for the nutraceutical industry, and you can see the herbal industry. And they have the methods or the procedure in check for the ginger raw material, for checking the raw material of the ginger, even if it is a powder or if it is a, you can say the raw, fresh ginger, to ensure the end product of the ginger is actually the quality one. So we need to ensure the quality of, let's say in the case of ginger, from the very beginning, starting from the cultivation, collection, processing, and then harvesting, and after that, storage, and then before preparation into the dosage form, it has to be gone. It has to go through various steps to ensure that it has a certificate of analysis which actually suits its quality profile. So to do that, we have certain procedures in place. So the first method to ensure that the ginger is the authentic one is the morphology. Okay, so in case of the authentic ginger, what could be the morphological structures which a person who is doing the analysis need to see? So in this experiment, we'll be looking at the morphology and microscopy and powder characteristics. These three important aspects of the ginger we will see. Of course, further doing the physicochemical analysis, doing the identification of various chemicals by TLC, or later by doing spectroscopic analysis. All these things are done in the industry. Okay, but for our experiment, we will limit this analysis until the powder characteristic. Okay, so if you look at the morphology of the ginger, you'll find is a flat piece. We will see when we do the practically, and shape is like a flat, laterally flat. And then if you look at the branches, that means there are some finger-like projections of varying with the size. I mean, it may be 1 centimeter, 2 centimeter, 3 centimeter. It may be even different in different varieties of the ginger. So it's always in the range. And then if you it has the branching, or we say finger-like projections, and then also have some striations, or that we will see with the help of magnifying glass. When you fracture it, the fracture will be fibrous, that we will see, and the color may be brown, maybe white. It depends the type of varieties, and even the processing, how it is being done. So sometimes we will see the ginger is brown color, sometimes we see it's towards whitish, towards yellowish. It also depends, like whether it is a fresh, stored, or if it is treated. Sometimes it is, in the adulteration, there are certain cases where it was found it was treated with some chemicals to remove the outer, to modify the outer appearance, okay, to look better, or maybe looks like very fresh and like whitish. So looking at the organoleptic, that means checking for the odor, color, taste, the aromatic. It has the aromatic odor and smell, or the taste is pungent because of the gingerol. And then looking at the 3S, we are going to see the outer surface, or the outer zone of the, or epidermal part of it. It will be mostly the Koch cells, and then parenchyma cells. We will see the starch grains with the treatment with the iodine and without iodine. So with the iodine, the starch grains will give blue color or bluish black color. And we'll see some vascular bundles, that means xylem and phloem, and also the oleoresins. That means a volatile oil with resin, and then xylem vessels. We may see even reticulate type. It may have even the other types of the xylem vessels. But most important part here is unlignified. That means it doesn't have the lignin into it. Okay? And then we'll see fibers, could be phloem fibers or xylem fibers. And then in the powder characteristics, we'll see the same components which is in microscopy, but when we make the powder of the ginger, definitely most of the parts will be broken, or you may not get an intact structure as you see into the TS. So we may see even xylem vessels, we may see even the starch grains or fibers into the powder characteristics as well. So these microscopic characteristics or powder characteristics, they are important because they are very specific to a particular species. So if there is an adulteration, and in the analysis, if you find there are some unwanted microscopic characteristics which are not in the genuine drug, then you can easily identify. Even though it looks similar from the appearance, okay, you can identify with the powder microscopy, because when ginger powder is made, we don't know whether the fresh material was the genuine drug or not. Okay, because when powder is made, we can only see with the powder microscopy. If you find the powder microscopy the the structures which is listed here, that means it is more towards the authentic. If you find some of the unusual characteristics which are not supposed to be in the ginger, then you can come out to the conclusion that it may be adulterated with some of the you can say the adulterants which are not supposed to be in the ginger. Okay, so if you look at even the importance of the ginger in Malaysia, the ginger has been included in the herbs which was selected under the National Key Economic Area Agriculture Project. They selected around 18 herbs, and later they brought it down to the 10 herbs, and in one of the herb is the ginger, Gingiber officinale, in Malaysia, for producing it as a value-added product. So you will see in Malaysia so many the herbal products along with the other Malaysian plants. Ginger is also there in the value-added product. So it's very, very important to learn about ginger, how to authenticate, how to identify the authentic ginger, especially with the morphology and microscopy. So we'll see in the experimental part. Yeah.
Okay, now we will do the morphology part of a ginger. So now you can see here some of the samples of the ginger. This is a fresh ginger, and as you can see that there are various sizes. Some are in small, some are in big. So that's why when we look at the morphological part of it, there will be range. Let's say when we see the length of it, it will be in the range, like width also in the range. Okay, so let's go and see one by one. All right, so first we have to see its shape. So if you look at this piece, the shape of this ginger is literally a flat piece. You can say it's a flat. Okay, it's a flat piece with the branches, finger-like branches. You can see here. So you can see here the branches here, finger-like branches. So it got branches and it is a flat piece. So we can even see the measurement later, like what is the size, especially the length, width, and we will see even the branches also in size. Okay, so let's see its length. So if we measure, its length is around, if you mark from here to here, is almost 27 centimeter in length. And if you look at its width here, it is around like 18 centimeter. And if you look at the projections, I mean the finger-like projections here, the length of this one is around 9 centimeter. This another finger-like projection. This another finger-like projection. It's around 7 centimeter. And you can see the another finger-like projection here also is around 4 centimeter. So you can say the range for finger-like projections is around 4 to 9 centimeter. And for the entire sample here, for this sample, you can say the length is around 27 centimeter and width is around 18 centimeter. So this is for particularly for this sample. So let me take another sample and then we see the size of it. And if you look at the size, I mean the length here, length is around 18 centimeter here. So this is number one. Number two is length is around 18 centimeter. And if you look at the width here, is around 4 centimeter. Okay, so let me take the another, which is the smallest sample which I have with me. So this is the smallest sample. So let's see, this smallest sample is actually is a broken part, so I'll choose the another one. Okay, so this is our sample 3, and here the length is around 14 centimeter and width is again around 5 centimeter. So I have taken 3 pieces, and if you look at these 3 pieces, the lowest length is around 14 centimeter. So as you can see in the sample number 3, and highest length is around 27 centimeter in sample number 1. So I can say the average length here, or you can say the minimum to maximum length, we had the sample of 14 to 27 centimeter of length, and width minimum is 4 centimeter here, and the maximum is 18 centimeter here. So we can say the width is around 4 to 18 centimeter. So we have these 3 samples with the range of length of you can say 14 to 20 centimeter and width of 4 to 18 centimeter, and they are the flat pieces, and one of the piece got a finger-like projection, and this finger-like projection got like you can see these projections have the range of the length of 4 to 9 centimeter. Okay, so this is about its size, how to check the size and shape of the ginger. So the next we will see about its organoleptic features. Alright, so now let's begin with its organoleptic feature. Organoleptic features means the features which can be identified with the help of sense organs. So let's say the first one is a color. So it is identified with the eyes. So we can see the color of this sample is yellowish, or you can say the pale yellow. But there are ginger which are whitish in color. Some of the gingers are buff in color. The color may be different in different varieties, and sometimes the color also is different because of the treatment. Like sometimes the ginger is treated with acid, sometimes it is treated with the lime. So you can find it like a lime ginger, or like a treated ginger with the acid. So the color will be a bit different in those samples. Alright, next is the odor. So when you smell this, of course to smell this we have to break it and then we have to smell. So when you smell, it is very what do you say, the agreeable, and the smell is aromatic. The aromatic smell of the ginger is because of the presence of the volatile oil. So since it contains the volatile oil, so you will have a aromatic odor or a smell. Now coming to the taste, and we all know the taste of the ginger is very pungent, and that depends on the pungent principle. Some of the ginger varieties have got this gingerol, which is more in amount, so it will be very much pungent. But some of the ginger, they have gingerol in the less amount, will be less pungent. So the pungency of the ginger also depends on the variety. So that's all about the color, odor and taste. Next we will see its some of the extra features with the help of a magnifying glass. Alright, so the next we are going to see the extra features. And as we know, this ginger is actually a rhizome, and there's a difference between roots and rhizomes. Rhizomes are actually the modified stems, and there is a presence of nodes and internodes. So you can see on the ginger there is a presence of nodes and internodes. So you can see here, this is one node, okay, this is the another node. So there is a distance in between two is called as internode. Okay, so distance between this node and this node is called as internode. But you can see at the nodes there's a very interesting, you can see the buds are coming out. So from the nodes the buds may grow if you keep it for a long time, or if you store it for a long time, the budding may take place at the nodes. So here in this ginger, since it is kept for a long time, so you can see the budding is coming up. And here in this case also you can see some of the buddings is coming up in this ginger as well. All right, so the next is a fracture. So we just fracture the ginger, or we can just break it and see what kind of fracture the ginger can give. So when you fracture the ginger, let's say when you break it, as you can see, this is fibrous. You can see the fracture surface is fibrous. Okay, you can see the fracture surface is fibrous, and you can see it very clearly with the help of magnifying glass. Okay, so the fracture surface is fibrous. Okay, and then next we can see is the striations. Striations means it's a longitudinal one. So it's like you will see the structure like this on the surface. This is a longitudinal striations. Okay, we call them a longitudinal striation. So maybe we can say one piece of the ginger, we can take a little bit of part of it, and then we can see the longitudinal. Okay, so you can see here, this one is longitudinal, the striations, line-like structures you can see here. Okay, so this is all about the morphology of the ginger. The most important part here to remember is the size, and you can say the measurement of the size of the ginger depends on the sample. So when you get a sample which is smaller than the sample which I use, the size may be a bit different, not the same as I mentioned here. All right, so in the next we will learn about the microscopy part. Yeah. For doing this experiment in lab, we need the materials. So for this experiment, we have the materials here. For the morphology, we have this magnifying glass, and for microscopy, we got these watch glasses. Then we got the slides, and we have these cover slips. We have the blades to cut the section, and we have some samples here. Like for today experiment, we have ginger, then we have this ginger powder, and then we have some chemicals here. So we are going to use only two chemicals here for microchemical test. One is iodine for presence of starch, and another one is sudanrate for the presence of volatile oil. And we can use the glycerine in case we want to just see the sample or TS without any microchemical test. Okay, to do this experiment, you need the sample, and in this case it is a ginger. So to have a successful microscopy of a ginger, we need to cut the transfer section of it. So the section can be cut in two ways. It can be a transfer section, it can be a longitudinal section. So when the sections are cut in a horizontal way, it is called as the transfer section. When the sections are cut in a longitudinal way, then it is called as longitudinal section. So in short, the transfer sections are also called as TS, and in the longitudinal sections are also called as LS. So this one is TS when it is cut horizontally, and when it is cut longitudinally, it is called as LS. So there is a difference between TS and LS. Now coming to the successful section, as I mentioned to you, the section has to be very thin. So if the section is very thin, we can see the tissues very clearly, and the size of the section also very important. So we need to make sure the size when we cut, it should be in a way that we can handle it properly and we can cut it properly. So for this section there is no problem, but when we have a section like leaf, or we have a very thin sections, for that purpose we need some kind of holder, like we can cut the potato and put the leaf in between, and we can cut the thin section. But in this case, since it is a rhizome, so we don't need a potato or any other holder to cut it, so we can straightforward go for cutting this section. To cut the section, we must hold the section between the thumb and a finger, okay, and the section should be large enough to be able to hold between the finger, okay. And then before we cut the section, we should also have some water on the blade so that the air bubble don't get entrapped when we cut the section. So let me make this sample in an appropriate size so that we can cut this and we can observe under the microscope. Obviously we cannot put the whole chunk of this under the microscope, so we have to cut it in such a way so that we can put it under the microscope and can see it. So we need to size it first properly. So we can make a small cube of it. But we have to ensure that the size should be not very small, or it should not be so short that we cannot hold in between the finger and thumb. Okay, so I think this size should be okay. So as you can see here, this size, this is a kind of a cube, okay, and we can also hold in between our finger very nicely, and we can also slice it horizontally. Okay, so now I'm going to slice this ginger into a very thin section, okay, and as you can see, this blade should be sharp enough to cut this section, and we should be able to properly hold it in between the fingers, and there should be some water on the blade so that the air should not entrap. And then, as the rule says that thinner the section, better the section, we have to slice it as thin as possible. So we have to slice it in such a way that it should be the very, very thin section. Okay, so I have just cut a little bit of it, and then with the help of the brush we have to transfer it into the watch glass filled with the distilled water. So I'm just transferring it with the help of the brush. Now you can see this section is in water and very, very thin section. So let me cut one more section, then we can put under the microscope. So again we can go for a very thin section. As I mentioned, the thinner the section, better is the observation under the microscope, because the structures will be very clear. Okay, so I've cut the section. Okay, this is a very thin section, and I'm going to put it with the help of brush. I'm going to put into the distilled water which is in the watch glass. Okay, so here you can cut as many as sections you want. Like we want section for microchemical tests with the iodine, and we want section to treat with the Sudan red tree for the volatile oil, and we need a section just for the purpose of observing without any chemical treatment. So let me cut one more, and then we can proceed. So I'm going to cut one more section here. As I mentioned, it should be very, very thin. Sharpness of the blade also plays a very important role. As you can see here, I have cut 3 sections. Okay, so one I will use for the normal observation without any chemical treatment. Then other 2 I will use with the chemical treatment, like using the microchemical test for iodine and Sudan red. Yeah. Alright, so we have 2 microchemical tests to carry out. So first one is the fluoroglycinol, and this fluoroglycinol is a chemical reagent, and this fluoroglycinol is used to stain the tissues which are lignified. So any of the tissue in the section which has the lignin will give pink to red color. Okay, so we need to prepare the reagent first. So we have a fluoroglycinol here, and we need to include fluoroglycinol and hydrochloric acid. Okay, we have a hydrochloric acid, concentrated hydrochloric acid. So we have to have the same ratio. So here in this watch glass, I am going to use, let's say, the 2 drops here, and I'm going to use the hydrochloric acid, concentrated hydrochloric acid, again 2 drops. So we have prepared our reagent for the fluorouracil. So we have added the equal amount of the fluorouracil and concentrated HCl. Okay, I have got 3 watch glasses. This one is having the sample, I mean the section just now we cut, okay, the transfer section. And this one, the next one is having the prepared reagent, fluorouracil with the concentrated HCl, just now we prepared, okay. And this third one is distilled water, plain distilled water. All right, so now we will take this section. I'm doing this procedure for staining purpose, because we are going to use the sample for the microchemical test also, right? So we need to prepare the we need to treat the sample with the reagent. Okay, so how to treat the sample with the reagent? So I'm going to transfer it with the help of brush. I have taken only one section from there. There are 3 sections here. I have taken one section from here. See, this is the section I have taken from here. Okay, and I'm transferring it into the fluorouracil with HCl. Okay, and I will leave it for minimum 2 to 3 minutes. And I have left it here for 2 to 3 minutes, and now fluorouracil will stain. It will stain any of the lignified material or any of the lignin containing tissue inside this TS. Okay, I will leave it for 2 to 3 minutes, and after that I will again transfer this one from here to water to remove any extra staining, because otherwise when you see, if you don't treat further with the water and directly see this sample under the microscope, the entire sample, if too much of lignin, will be fully pink or red, and then we can't make out the characteristics properly. So to avoid that, we need to further treat this one with the distilled water, so that the excessive staining or extra staining is washed off, and then our sample is properly stained with the fluorouracil, if at all having any lignin, and then we can see it under the microscope. Okay, so now after 2 minutes, we are transferring this section which was under the fluorouracil to the distilled water. Okay, so finally whatever the extra staining is there, that will be washed off in this distilled water treatment, and then further we will take it for observation under the microscope. Alright, so next is the mounting process. For mounting process, we need a clean glass microslide. So we have this clean glass microslide, and we need samples. So we already cut a few of the ginger transfer sections here. So first we will transfer this section onto the microslide. So we need a brush, we need a needle, and we need the forceps, and also a water with the dropper. Okay, so let me take this one of the TS, okay, and then with the help of the brush, let me mount it on the microslide. Okay, and then we need to cover it with the cover slip, but before that we need to put a drop of water so that our sample gets submerged in water. We can even use the glycerine in case we are going to use this TS for observation for long hours, maybe for a day or two. But since I'm only using it for a few minutes or a few hours, so I'm going to use the water here. Okay, so I will just put 1 or 2 drops of water so that the sample gets submerged in water. All right, and then later I will use this cover slip with the help of forceps to put it onto the sample here. Okay, on the TS. So you can see here, and the most important part here is there shouldn't be any kind of air bubble. And we can add some more drops of water from side so that there shouldn't be any air bubble. And later we can remove the extra water with the help of a paper, tissue paper or blotting paper. So as you can see here, there is no bubble. Okay, there is no air bubble, and we can remove the extra water with the help of the tissue paper. So as you can see here, that there is no air bubble in our sample. Okay, you can see there is no air bubble, so our observation will be clear. Okay, likewise we are going to take this TS, which was already treated with the fluoroglycinol and HCl. Okay, so we kept it in the distilled water, okay, for removing the extra staining, and from there we can take it. All right, so now I have kept it on the microslide, okay, glass microslide, and then we can add 1 or 2 drops of water, and then we can use the cover slips, okay, to cover this. So now you can see there is no air bubble. You can see there is no air bubble, and now this slide is also ready for observation under the microscope. All right, so next we are going to have the staining with the iodine. Okay, so we are going to use the one more sample, and we are going to put it on the microslide, and then we have to add a drop of iodine. And this one will give us the clue if at all our sample contains the starch, because the iodine is used for detection of the starch. Okay, so we just put 1 drop here, and then we are going to put the cover slip here, the same way, and again we make sure that there shouldn't be any air bubble. we can remove the extra staining with the help of the paper. Since we can see there is a one bubble here, so we need to lift it again. See, with the help of needle we have to lift again, and we need to make sure this air bubble goes away. Okay, but still we can see this one air bubble here inside the sample. You can see here. So again we have to make sure, yeah, this bubble is gone now. So there's no air bubble inside the sample. You can see there's a one air bubble here again. This is nearby sample. Okay, gone. And we can remove the extra staining. So now, as you can see, this sample doesn't have any air bubble. So I think this sample treated with the iodine is now ready for observation under microscope. Okay, so the next we have to do the staining with the Sudan rate for determination of volatile oil. If the sample has any volatile oil, we will get the red color when we treat with the Sudan rate. Since our sample is very thin, okay, so let me put the drop of the Sudan rate, okay, and then we have to put the cover slip. Later we can remove the extra staining with the help of tissue paper or blotting paper. But anyway, you can see there is no air bubble inside the sample. Okay, so this is now ready for observation under the microscope. Later I'll remove the extra staining from here. Okay, for powder microscopy we have to take powder. Okay, so we need to use the brush to mount the powder on a glass slide. So we have to take a fresh clean micro glass slide, and then we have to take a pinch of powder with the help of brush, and then we can put it onto the glass slide, and we can even scatter with the help of the cover slip if you want to scatter, so that the structures are more clear and they are not very condensed when we see in the powder microscopy. Okay, so with the help of the forceps, we can put the cover slip here, and this one is without any chemical treatment. Okay, this is without any chemical treatment. So this we are going to observe without any chemical treatment. Likewise, we are going to prepare for chemical treatment. Okay, so we are going to do the microchemical test on the powder, ginger powder. So we are going to take a pinch of the ginger powder on each slide, because we are going to do the 3 microchemical tests. so we are going to use the fluorouracil with the hydrochloric acid. We're going to put a 1 drop of the fluorouracil here and 1 drop of the hydrochloric acid, concentrated hydrochloric acid. All right, in case if any lignified tissue is there. Okay, and this next treatment is with the iodine for identification of any of the starch compounds or maybe a starch grain. Okay, so we just put a 1 drop here on the powder. Okay, let me put 1 more drop. Later I'll put the cover slip. Okay, and here in this case the sudanrate, in case if we have any volatile oil, which are highly unlikely because volatile oil normally when we prepare the powder normally will evaporate during the processing. So later, now I'll put the cover slip on these 3. So I'm going to put the cover slip on the first one with the fluorouracil and the hydrochloric acid. Okay, this is for powder, and this one I'm going to put it for the iodine. Okay, I think I need to put some more iodine here. And then the next one, I'm going to use 1 more glass cover slip. Okay, so put it onto the sample treated with the sudanrate. So now these 3 powdered microscopy slides treated with 3 different chemicals. The first one with the fluorouracil and hydrochloric acid, concentrated hydrochloric acid, for the detection if any lignified compounds are there in the powder. The second one treated with the iodine to detect if any starch compounds or starch grains are there, and the third one treated with the sudanrate 3 for detection of any of the volatile oil component. All right. Alright, so the next is we are going to use the compound microscope attached with the computer so that we can see the images on the screen. So we can say it's a kind of microscope attached with the camera, okay, so that we can see the microscopic image on the computer. Alright, so this compound microscope is made up of the eyepiece lens and the objective lens. So we can see this is the eyepiece, okay, and then we have this objective lens here. So we got this 10x, then we have got this 40x, then we have got this 100x. So we are going to use 10x and the 40x. 10x is with the, you can see the yellow coding, and this 40x is with the blue coding, blue color coding. Okay, and then you can say this is the stage, okay, and then we have this diaphragm here, okay, below, so that we can adjust for the better resolution. All right, and then this kind of microscope is very good in comparison to the simple microscope, wherein only the eyepiece lens is there. There is no objective lens in the simple microscope, so the magnification here will be higher. Let's say if this eyepiece lens is of 10x, if this eyepiece lens is of 10x and this objective lens is 10x, then 10 multiply 10, that means we are looking at the object 100 times more than its original size. And when you are using this 40x, and if this eyepiece is 10x and this is 40x, that means we are looking at the object 10 multiply 40, 400 times bigger than its original size. So that's the importance of using this compound microscope for the identification of the micro structures in the transfer section. All right, so now as you can see here, I have put the uh, TS sample here inside, and you can see the image over there. Okay, you can see the image over there. So we can see here, you can see the yellow color here. That means there is a volatile oil present here. You can see this, as I'm mentioning here, there is a presence of vascular bundles here. All right, and then we can see so many parenchymatous cells. Okay, so we will capture the photos. Okay, we will capture the photos, and then later I will explain to you the each structure in detail in these images. Okay, so you can see here, now I have put the sample, and now you can see the image here, and you can see the iodine treated sample, and you can see all these small starch grains which are blue color, bluish black color.
All right, so let's have a look on the results of the transfer section and the powder microscopy. So before we proceed with the results, just to remind that we have done the section chemical treatment with three main chemicals. One is the fluorouracil, another one is the iodine, and the sudanrate. We treated with the fluorouracil to check if there's a presence of lignin. We treated with the iodine to check for the presence of starch, and we treated with sudanrate to check the presence of volatile oil. All right, so we will see one by one. So first we see the results of without any chemical treatment. That means we cut the TS and just put under the microscope and see how the results appear. Okay, so this picture shows clearly that there is a presence of the vascular bundle. There is a presence of the oil cells with the resin. So as you can see here, these cells are the oleoresin cells. Okay, you can see here, these are the oleoresin cells, and these are all vascular bundles. This is again a oleoresin cell, and this is the vascular bundle. All these circle marked circle is for vascular bundle. Even in fact, you can see this one here also vascular bundle. This is also vascular bundle. Vascular bundles are actually they are made up of the xylem and phloem, and as we know, the role of the xylem and phloem, the xylem tissues, they are actually transport water and nutrients from root to the different part of the plant, and plays an important role in the structural support in the stem mainly. But here in this case, ours is a rhizome, so of course it also has the xylem present in it. And then the phloem tissues, they transport the organic compounds from the side of photosynthesis. So we know the photosynthesis produce the sugar, and then from there it is transported to the different part of the plant through the phloem. So this vascular bundle is made up of the xylem and phloem. So later we will see the individual part of the xylem and phloem component. So now this is the larger picture of what we seen in the previous picture here. You can see these are the starch grains, as you can see, and these starch grains looks like a bit transparent because we have not treated with the iodine, otherwise they will be colorful. And here you can see this is the vascular bundle, the larger picture. Okay, you can see the xylem. Okay, the xylems are normally larger than the phloems. You can see the phloems are like the bunch of grapes here. And then if you see the type of the vascular bundle which is present in the ginger is a collateral type of vascular bundles. There are different type of vascular bundles like conjoint vascular bundle, collateral vascular bundle. But in the collateral vascular bundle here also there are two types, open and closed. And here in this vascular bundle, since it is a collateral type, that means the xylem is at the the phloem is on the outer part than the xylem on the same axis. So that is for the you can say the further learning. If you want to see the detailed structure of each component of the vascular bundle. So next we will see is the again some of the other structures without any chemical treatment. As I mentioned that this is the outer surface of the cork cells, and there is of course there is an inner cork as well. So this is the outer cork of the TS. So that shows that it is not the Jamaican ginger variety. And then if you can see here is the we have done even the measurement here. So you can see the vascular bundle here with the xylem and phloem. The length of this vascular bundle is around 43.363 micrometer, or in short, you can say microns. And the oleoresin cell, the size is here 13.485 microns. And here you can see the size of the parenchyma cells are around 13. One of the parenchyma cells is around 13.442 micron. Okay, here next is the transverse section of the ginger treated with the fluorouracil. Of course, we taken the equal amount of the fluorouracil and the concentrated HCl, and then we dip the section into it for 2 minutes and wash with the water, and later we mount. And here is the result of that section. And there is no pink color or pink to red color in this, you can say, picture that shows that the sample what we have taken doesn't have the lignified tissues. So that also depends whether the tissue is new or old. And since in this part we don't see any lignified tissues, that there's no pink to red color, so we can say the lignin is absent. Okay, so the fluorouracil actually react with the lignin and give pink to red color. And in this, we don't see any pink to red color. So our conclusion is the lignin is absent, but we take the advantage of this picture, and then we can see the other structures which are present. So you can see the oleoresin cells are obviously seen here, and these are polygonal. That means various sizes of the parenchyma cells. You can see some cells are bigger in size, some are small in size. So we say they are of different size polygonal parenchyma cells. And here you can see these are the xylem vessels. Okay. All right. And this one is under the 10x view, and this is the 40x. That means even the more larger, the resolution. As you see 40x, that means 10x is eyepiece, 40x is objective lens. That means we are looking at this picture 400 times bigger than the original size. So you can see here, this is the vascular, the xylem vessel bundle. Xylem vessels are actually combined together in a kind of bundle form. And then you can see this, the length here is around 272.161 micron. That's the size of this vascular, the xylem vessel bundle what we have taken here. But of course, the lignin is absent. That means there is no red to pink color here. That means the lignin is absent. So we can say the xylem vessels are not lignified. Now again, this is the one more picture of the TS of the ginger treated with the fluorouracil, but with the 40x view. That means the larger view, and you can see the clear picture of the phloem and xylem. And we can see these the phloem. You can see this is the phloem, and this is again a phloem. As I mentioned before, that they are outer than the xylem. They are outer than they are in the outer or exarch, we say, in this kind of a vascular bundle. And here you can see again, you can see presence of the oleoresin cells. And we have a starch grains as well. And you can see these are starch grains are also measured, like one is a 4 micron, another one is a 6.675, another one is 2.60. So the various sizes of these starch grains here. Okay, so the next picture here, this is of TS treated with the iodine. So what's the purpose of iodine? We use in this experiment to check if the starch is present. So what would be the result when we treat the sample or TS with the iodine? The iodine will change the color of the structures which contain starch into bluish black color. So as you can see in this entire picture, you can see this, all this blackish color is actually the starch. That means you can see there are so many tiny structures with the black dots here. They are all starch grains. So that means this ginger have a abundant amount of starch. So much of starch grains are present in the ginger. And you can see these are the vessels. You can see these are the bundle of the xylem vessels again. And again, these are the vascular bundles. You can see here, these are the vascular bundles. And of course, this is the outer, you can say the cork cells. Okay. So next is the treatment with the iodine. Again, for this TS, this is the 10x view. And here you can see this is again a vascular bundle, but our purpose in this structure is to see whether the starch grains are present or not. And as we can clearly see, these starch grains are present because they are very tiny structures shown with the bluish black color. Okay, so these are all starch grains, which you can see small tiny structures with the bluish black color. So we can say the starch grains are present because they give color with the iodine. Again, this is the another view of the same results, wherein we can see the starch grains are present. Okay, you can see the starch grains are present. Okay, this is again the xylem vessel bundles here. And you can see this one is the reticulated xylem vessel type. Even it can have a different type of you can say the xylem vessels as well. It's not that it has to has only the reticulated type. We have identified one of the type is reticulated. Of course, it may have an annular or spiral type of the xylem vessels as well. So it may have an annual and also a spiral type, but we have identified here is the reticulate type of the xylem vessel. So there are different types of the xylem vessels as well. So the conclusion of this picture is the starch grains are present because when we treat with the iodine, it shows the tiny structure of the starch grains with the bluish black color. Now, here this picture is taken in 40x. The purpose of this picture was to see the starch grains structures in the larger view. At the same time, we found that there is the oleoresin cells here, and which is very much clear. It contains the volatile oil and resin. That shows that our sample is not exhausted with the volatile oil. So you must have studied in the volatile oil chapter that most of the plants, the adulteration in them is like they are being exhausted. That means the volatile oil component of those plants are actually exhausted, and then they are sold in the market without the presence of the volatile oil by adding the artificial aroma and all. So here in this case, as you can see, the volatile oil is present. That means this is not an adulterated sample because it shows that the oleoresin cells contains the volatile oil. Now, next is the treatment with the sudanrate 3, and the purpose of this reagent is to identify if there is a presence of the volatile oil, and this actually corroborates our previous finding of the volatile oil cells, that when we treat, it shows the the red color. Okay, when there is a presence of the volatile oil, you can see there are so many structures here. There are so many tiny red color structures. That means when you slice the TS, or when you cut the the ginger with the blade, cause the volatile oil, when the cells are ruptured, it will spill over. Okay, and when you treat this, definitely you will get the picture wherein you will see the structures like this treatment treated with the sudanrate reagent. However, we can see there are presence of the xylem vessels here, then there is a presence of phloem, and again a very clear picture of the reticulated type of xylem vessels. But the conclusion of this slide is the volatile oil is present because it shows the red color with the sudanrate, the reagent. Now next is the powder microscopy. The results in the transfer sections were very obvious because the structures were intact. Okay, the structures were intact. You just put the reagent and then you can observe under the microscope, and the results are very clear. But in the powder microscopy, the problem is when you crush the crude drug. That means we to make a powder, we have to dry the ginger first, and after drying the ginger, we have to put into the blender, and then we have to make the powder. So most of the structures, the microscopic structures, are broken when it is made into the form of powder. So in the powder, we have to identify the structure, and it's very tricky because sometimes the intact structure is broken in many pieces or many parts, and then we have to identify it. So it's a bit tricky, but it's very, very important. Why this powder microscopy is very, very important? Because if there is any kind of adulteration in the crude drug or in the fresh drug, you can identify. But if there is any kind of adulteration in the powder, it is very hard to identify the adulteration in the powder until unless we do the powder microscopy. So that's the reason the powder microscopy plays a very, very important role in identifying the adulteration in the powders. So here also we will see some of the images without any chemical treatment and with fluorouracil, and of course with iodine and with the sudanrate. So this one is without any chemical treatment. As I mentioned to you, you will not see the intact structures as we've seen in the previous slides, but here you will see these are the broken structures, broken parts, and but here it's very clear that it shows a kind of a fiber here. Okay, it's 83.487 micron in the size, and here in this it's again corroborate the presence of the cork cells. So you can see these are the cork cells, okay, which are in the outer or inner surface of the ginger. And here again you can see this one is a xylem. This one is a xylem, but it's a 40x view of the xylem, so it's very clear. It's a you can see here this one is more of a reticulate type of xylem. And again, this is again the powder treated with the fluorouracil, and you can see this is a similar picture having the cork cells, and then these are some of the broken parts, which could be phloem fiber, or it could be any other structure, but very hard to identify because it is just broken part. And here it is the xylem. Okay, you can see clearly this is xylem, and these are the xylem vessels. Okay, these are the xylem vessels. And now next is the ginger powder treated with the iodine. So here you can see, you will find so many tiny structures which are like black dots, or the small black side structures, and all these are actually starch grains. So that means the ginger powder also shows that the powder is taken from the authentic sample because it contains the starch. So the starch grains are present in the powder of the ginger. Now this is the another picture of the powder microscopy wherein we have taken the measurement of these starch grains, of course with the larger view 40x, and you can see there are so many of them, and you can see the sizes. I mean the other structures are also surrounded, but the more obvious one is the starch grains with the size. Next is the ginger powder showing the presence of the xylem vessel, and as I mentioned that this is again a bigger picture in the 40x, and it's very clear. Even you can see the size is mentioned 42.802 micron, and it's a clear picture of the reticulate type of the xylem vessel, which we seen in our intact pictures or intact structures of the transfer section of the ginger. Now again, this is again a xylem vessel, but you can see nearby these tiny structures of the starch grains again. Now this is the ginger powder treatment with the sudanrate. So however, you can see a very small structures. You can see very small dot-like structures with the brownish red, but very less. So since this powder is prepared after crushing this dried ginger, that means when you are doing, I mean, when you are when you are putting this dry ginger in the blender, because of the blender heat, when you are making the powder, there is a high chance that the volatile oil cannot stand that temperature and will be evaporated. So in the powder, if it is blended at the high speed and with the temperature, definitely the volatile oil presence will be questionable. So here also you can see in the powder the volatile oil presence is very minimal, or if it is stored for long time, definitely you will not see much of the volatile oil in powder. So now this was all about our results of um, the TS of the ginger and the powder microscopy of the ginger. Now, how to write the report? Okay, for writing the report, you need to write of course, you need to write the aim or the objective of the experiment. Then you need to write the requirement of the experiment. Then you need to write the background of the experiment, um, like why we need to do this experiment. Then why you need to even write the procedure for the morphology and microscopy, and then you need to write the observation. Okay, so in the observation you need to draw the structures, okay, given in the practical manual. You need to write draw the structures, okay, and of course you have to uh, put the observation of these results here. All right, so uh, if you want, if you have a ginger sample, okay, it's voluntarily, not compulsory. If you have a ginger sample at your home, if you want to put the observation of microscopy, it's fine, because that doesn't need any kind of the instruments, but it's all up to you. But if you want to put the results which I have done in this experiment, you can put, it's up to you. Okay, then next is the microchemical test here. Microchemical test, I have already shown the results. We need to put the observation and characteristics of these results. Okay, treated with the fluorouracil, concentrated HCl. Observation here in this case, observation that there was no red to pink to red color structure. So you can say in characteristic, you can say the lignin is absent. Then in iodine, observation was the bluish black color, the tiny structures were observed. Characteristic is, you can say, the starch grains were present. If you want to write for the, you can say the starch grains, they are actually the simple starch grains, because if you look at the starch grains also, there are various types. So it's a simple starch grains. And then next is the Sudan-Red 3. In the Sudan-Red 3 also, in the observation, you can write the structure shows the presence, the structure showed the red color after treatment with the Sudan-Red 3, and characteristic, you can see the volatile oil may be present. Okay, so when it comes to the report writing, you need to write in the report that it has to be the report has to be in the passive voice, okay, past tense, that the morphology, microscopy, and powder characteristic of the ginger was studied, and the result shows the sample is authentic based on the observation into the morphology and microscopy. Okay, and if you want, you can put the presence of the let's say the starch grains and presence of the volatile oil to make it like more uh, substantial report. And then coming to the discussion, in the discussion you need to write uh, what are the things we use in this experiment. Let's say the fluorouracil, you can write the chemical structure of fluorouracil, and then you can write how it react with the lignin and how it gives the color. Likewise, you can write about the starch grains, type of the starch grains found in the, let's say, maize or rice or potato. There are different type of starch grains, and then you can write about like what kind of starch grains are present in ginger. All right, and later there are the questions here, so you need to answer those questions in the report. So that's all on how to write a report on the ginger. So I hope this entire video will be helpful for you to uh, understand the experiment on the ginger. And if you have any question, you can always send an email, or you can contact me if you have any question on this experiment. Yeah, thank you.